just ignore the beeping sound. And Lizelle, are you starting the recording now?
Yes, I've started recording. Thank you.
Okay, great. Thank you. Okay, great. So we're here today to talk about preparing environmental samples for next generation sequencing. And my guest today on this presentation is Dr. Henry Ogola. Dr. Ogola is a senior lecturer in applied microbiology at the School of Agricultural and Food Science in Kenya, and he got his PhD in bioresources sciences from Totori University in Japan. He's very much interested in microbial metagenomics, oecology, bioprospecting, and genome mining for identifying and utilizing industrially important enzymes. So he's very much hands-on on more or less everything that we're going to be covering today. So please, if you have technical questions that have to do with preparing environmental samples for NGS, do not hesitate to ask. Dr. Ogola, thank you again for making yourself available. Okay, so this morning, my presentation is going to go through looking at some of the ways that next generation sequencing is currently being applied for environmental samples. What are the sequencing methods, and a brief overview of what your sample requirements are for NGS, especially for the Illumina platforms. We would also look at some of the challenges that you get to face when you are using or planning to use environmental samples for NGS and our proposed solutions. In the later part of the presentation, I'll go through how MN Makeri Nagel, who is one of our suppliers, principal suppliers for nucleic acid purification products, addresses these challenges specifically in categories of soil, food, stool, and water or air samples. And we'll have some concluding remarks at the end. So, what it is relevant in this presentation is that when you get environmental samples for next generation sequencing, you are usually looking at utilizing that data for one of these 5 categories. Most people or some people look at employing metagenomics for improving the understanding of nutrient cycling and plant disease detection, as well as optimizing farming practices to improve crop health and yield, and this will be in agriculture. Now, when we look at biofuel, we know that biofuel is produced by the decomposition of the biomass by microbial communities. And so with NGS, we are able to gain new insights into the functions of these associated microbial communities and how they can be optimized for large-scale production of biofuels. Now, in ecology, NGS is currently offering one of the most valuable source of information for developing an aggregate understanding of the key processes that affect our environment. So NGS is relevant in this sector as well. And in a related area of study, looking at environmental remediation, when you use NGS to evaluate and eliminate pollutants in the ecosystem, you can base your NGS, or you can use NGS in order to study how pollutants interact with microbial communities, and then evaluate that and make a case for the elimination of these pollutants from the environment. And last but not least, looking at studying microbial communities that directly impact health. Most of the time, people look at gut microbe species, and also just in food safety as well. You can use NGS to differentiate between infectious and non-infectious diseases. And another growing area of application is tracking antimicrobial, antibiotic resistance genes in the environment. Now, what are some of the methods that are employed in next generation sequencing to achieve those goals within the study areas highlighted in the previous slide? So initially, or one of the categories is you can employ whole genome sequencing either from isolates or in non-culturable microbial communities, or you can use PCR to amplify and sequence certain targeted genes that allow differentiation of variable species or genera within a group of organisms. You can also look at targeted sequencing based on probes that Illumina particularly offers. And there is some panels that are for microbiology specifically, looking at antibiotic resistance genes, and some in the near future, there's also talks about you being able to customize panels for specific genes that you're interested in. Another area of methods is using environmental DNA for metabarcoding of eukaryotic species that are found in the environment. And this is usually done by targeting genes like the mitochondrial cytochrome oxidase 1 or 18S ribosomal RNA. Gene expression profiling can be done by metatranscriptomics, as well as looking at quantifying translation of certain genes by meta-RiboSeq. This is a very recent method that is also being explored at the moment using NGS. So the idea here is just to highlight these methods, but in week 4 and 5 of this series, Maria and the team will go in depth into these different methods as they apply on the different platforms that are offered by Illumina. There you will see what are the different applications, what are the different methods that can be performed on each of the benchtop instruments, and what kind of output you can employ for those. So can I just take a moment now to ask everyone if you can go into the chat box and scroll all the way to the top in the chat, and you will see there's a poll that I put in there. If you can answer the first question for me, which just wants to know what is the source of your nucleic acid. So if you're working from the environment, we know that this session was going to focus on samples from water, food, stool, soil, air. So if you can just enter, just fill in the poll and submit your vote. I would like to see at the end what the representation is like in terms of the different sample types that can come from the environment. And while we are on it, you can also just go to the second question and tell us a little bit what area it is that you would like to apply NGS in. So we saw already that there are 5 major areas that can be used for or where NGS can apply: agriculture, biofuel, ecology, environmental remediation, and medical studies. If you can just take a minute just to take a vote on the poll for the second question as well. Thank you. Okay, so once we have our samples, I'm not going to elaborate on sampling and experimental design, but definitely you want to consider that in terms of what the goal of your study will be, so that when you do have the results, you're able to infer that back onto the source of your samples. Okay, so it's an entire workflow, and whatever you get during your data analysis will be impacted by each of these steps. So from the sampling, extraction, amplification, and through the sequencing process. And for each of the different methods, there's different recommendations or requirements for the Illumina library preps in terms of the quantity that you will need of DNA or RNA, and as well the purity. This we'll go more into detail next week. On Friday, we'll be looking at quality control and how you can make sure that you check that your samples indeed fulfill the requirements for the Illumina sequencing platforms. So this is just keeping in mind all of that. Now, environmental samples come with a variety and not small challenges. Like, you have really impressive challenges. First of all, most of the times you have to collect your samples in the field and then manage to preserve them for processing in the lab. And we're talking about finicky nucleic acids like RNA that will degrade because of the abundance of RNases in the environment. So different samples come with varied challenges, and even within the same group of samples. So if you're looking for example, you have different types. Every other type of soil comes with challenge, and the challenges range from chemical right through to physical, and it's not consistent. So you have variable nucleic acid content at different times. And because you have to have a representative idea of what is happening in your area of sampling, there's usually a need for collecting lots of samples. And so there might be a need to consider automation during your processing of your samples. So those are some of the challenges, and I hope that I've touched the challenges that you face as the researchers in this field. If I haven't, and if I've omitted something, please feel free to share at the end of the presentation. It will be very useful. Okay, so when we're looking at now providing some solutions for the challenges that come along with environmental samples. First of all, in the case of remote sampling, you either want to snap freeze your samples to preserve the integrity of and the yield, to optimize yield eventually for processing, or you want to use a stabilization buffer. So from our providers, we have this cryopod carrier that provides safe, reliable, and portable cryogenic transport. So you can actually transport samples at minus or under minus 150 degrees, and this over 4 hours. So this allows you to be able to sample and carry your samples in the appropriate temperature to the lab. And this is particularly relevant, I think, for RNA, because we know that at very low temperatures, the RNases will not be as active to carry out the degradation of the RNA. And this this cryopod carrier displays temperature, date, as well as time, and it features audible and visual alarms. Another solution, which would then be for stabilizing your samples as an alternative, is to use the NucleoProtect RNA from Macarinagel. It's similar in efficacy, if not more so, to its competitors like your DNA RNA Shield or your RNA Later. So it's the product that is produced by MN for that purpose. Something else to put out there is that sometimes it's not about remote sampling, but sometimes it's about sampling in order to send your samples away for the actual extraction and then sequencing. In that case, maybe you also want to consider lyophilizing, that is freeze drying your samples, which allows you to cut down on cost that comes with cold transporting of samples for long distances. So that's just something out there. We do have a range of freeze dryers from Martin Crease that we also able to support you with. So if you already have your samples in the lab, something else that we would want to recommend are just some best practices that you can also include in your workflow to improve the yield and reduce contamination during the preparation of your nucleic acids. So the first thing is you want to, obviously, if you're going to be producing or isolating RNA, you want to keep your space and labware clean of all RNAs. And we have a solution that is non-alkaline, non-corrosive, and non-carcinogenic, which is RNase off from Takara. In another related work, if you're going to be doing any PCR work, there's a DNA off that also is from Takara that will allow you to eliminate potential DNA contamination of your PCR solutions or products. And finally, we also recommend that if you really want to take it to the next level, you choose to work ice-free, because sometimes, especially when you're trying to isolate RNA, you could get contamination or RNase contamination, not from yourself or anything else, but coming from the ice. So if you do ice-free work, you can limit contamination, and from left to right, you're increasing the reproduction of or the consistency of your method, as well as temperature distribution in the samples, which allows you then to have a more controlled workflow, so that every time, or if irrespective of who is going to be doing the work, we have some reproducible results. So this is just technologies that are out there. I'm not sure if you are aware of them, but they're out there, and we can assist you with that, just to optimize and limit some of those things that really can be limited. And we can then look at, when you are indeed doing your isolation, what are also those intrinsic solutions that we can provide you to optimize yield, limit contamination, and improve the quality of your nucleic acids. And in this area, we focus on three targets that are involved in sample pre-treatment. We look at sample disruption, a chemical follow-up to accompany the sample disruption to optimize lysis and yield. And then as a third strategy, we also able to provide solutions for removal of inhibitors. We are aware that environmental samples come with an abundance of inhibitors. And so there's strategies out there to target and eliminate those inhibitors so that our NGS and PCR downstream reactions are more successful. So as a first strategy in sample disruption, you will see in the later slides that most of the kits will involve a bead tube. These are tubes that come preloaded with beads, specific beads for specific types of tubes. We also have the compatible bead holders, bead tube holders, and they are compatible with vortexes that you can then use for manual homogenization of your samples. And in this slide, we just want to highlight that there are different types of beads that target different types of samples. So it's different materials and actual diameters of the bead. So there's some science and engineering that goes into the design of these homogenization beads. And they come for the environmental samples, most of them will need the type A beads, and they come already included in the stool and soil kits. And for the water kits, you can buy them separately. Okay, so as a follow-up to your physical sample disruption, we also want to highlight that with the McCurry-Nagel kits, there is an optimization of the buffer chemistry just for specific samples to really make sure that you get the most out of your sample. So different buffers would target efficient nucleic acid extraction or minimal releasing of inhibitors. They can also assist with the lysis, as well as some kits come with alternative lysis systems to address the diversity within a certain category of samples. Right, and finally, as a third strategy in the pre-treatment of your samples, you also want to look at strategies for removing inhibitors. And for this, there is you have columns and precipitation strategies, as well as wash steps, and these are differentially employed in different kits for specific characteristics of the sample type. So what are some of these inhibitors that we find in environmental samples? You see here they range from the humic acid to complex polysaccharides. And really why we worried about them is that they play a significant role in failure of your polymerase chain reaction type experiments, which definitely is very much at the heart of next generation sequencing. So we want to really focus on getting our samples rid of these inhibitors. Right. So at this point, I also just want to ask you again, if you could scroll. So I don't know if it's working in the chat box, but it's really at the top. So if you can just hover, go into the chat, hover all the way up. It's the first thing on the chat box. You just go to question 3 and tell me, have you used McCurry-Nagel kits? And can you attest to all the things I've said about pre-treatment strategies to address your different sample types? So it's just a yes or a no if you've used McCurry-Nagel kits. You can take a minute just to do that, please. Okay, so at this stage of the presentation, I'm going to be going into each of the different sample types that we find in the environment. In the first session this morning, Dr. Thomas of MN had a session looking specifically at plants, so I won't be talking about plants. And if you were wondering what HTL here is hydrothermal liquefaction of biomass. Also, we won't be going into that, but it just forms part of the circle of life. For DNA extraction from soil, we have the NucleoSpin Soil Kit. Like I said, it addresses sample disruption by including bead tubes of type A. It is based on silica membrane technology, and it is able to efficiently isolate DNA from a variety of soil types. And just as a highlight, there will be a magnetic bead-based kit in the nearest future from Nagel as well. And so the bead tube type A that comes in all our soil kits, they are very efficient in grinding the soil, as well as grinding in impacting the soil to lyse and release the nucleic acids, as well as grinding the microbes for lysis purposes. And they come as well with four alternative, the kits come with 4 alternative lysis systems, which ensures that you always get the highest yield from your specific sample type within the range of soils. So what that means is that there's always, the kits come with a buffer, SL1 and SL2, so there are 2 types of buffers, and they can be used simply, or you can add a chemical additive, which is the enhancer SX, that will allow even better, for some cases, better yield of your DNA. And also there is complete removal of PCR inhibitors, and your DNA is ready for NGS or downstream applications. There's no need for any further cleanup. So this is just a few gel images showing how using the alternative lysis systems, you're able to optimize yield. So for example, in potting soil, we can see that if we combine the second buffer with the enhancer, we tend to get a better yield. Whereas for river sediment, it will just be using the first buffer alone. So this, you can then look at what will be the best system, lysis buffer system for you or for your sample type. And when you compare the highest yield from the optimized lysis system with competitor kits, MN always gets a better yield. And not just the yield, but also in terms of removal of PCR inhibitors. The MN kits efficiently remove PCR inhibitors, and you see there's always consistent amplification, but again, relying on optimizing the right lysis system for you. And our guest in this presentation or during this session, Dr. Ogola, is a witness to the efficiency of the NucleoSpin Soil Kit. His group was initially using the Zymo Research DNA extraction kits, but then they used to experience failed extraction of DNA in samples that were rich in heavy metal concentrations. And these are not my words, these are Dr. Ogola's words, and I really thank him for his feedback and for sharing this with us. And in Dr. Ogola's own words, the kit gave good performance in extracting DNA from rhizospheric soil samples, even in plants that grow in acid mine drainage environment, a case that was not successful with the Zymo kit. And he says you can rely on the kit for high yield and quality DNA, even for challenging environmental samples. So we have, for RNA extraction from soil, an alternative technology that is employed just because in your soil, we are aware that the amount of DNA always outweighs the amount of RNA. And so you want to really optimize the yield of your RNA, which is why the kits employ the anion exchange technology as opposed to the silica membrane technology in the previous kit. And we always get high yield and for diverse types of soil, and your RNA is always within the purity requirements for NGS. Also, it's very easy to handle and it's very fast. And you can use a DNA set for this kit to have isolation of both RNA and DNA. Now, for the second category of environmental samples, you can isolate DNA from food using the NucleoSpin food kit. And this kit equally employs a special lysis buffer, like was highlighted previously, that allows extraction of DNA from various types of food and feed samples, as well as ensuring complete removal of inhibitors. And generally, the interest here is identification of pathogens, also differentiating between infectious pathogens and non-infectious strains, looking also at genetically modified organisms, or if you are doing halal food testing. So that is usually the interest for employing the food kit. There's also a magnetic bead-based format of a kit that will allow you to then automate extraction of DNA from large sample volumes or number of samples, allowing high throughput and within reasonable times. And you can see here how if you're using the NucleoSpin food kit, you're able to efficiently get high quality DNA from across several different types of food, even those with complex matrices. And this DNA is then able to undergo PCR reactions, even for quantitative PCR. For stool, we have a NucleoSpin DNA stool kit. So generally, every time you see a NucleoSpin, every time you see the spin here, it is a silica membrane technology that is being employed. And these kits come again already with your ceramic beads for homogenization of your samples. And they can be used both for human and animal stool samples. And you also get complete removal of PCR inhibitors. So those strategies that we looked at initially for pre-treatment, you can see now that in each of the kits, there is indeed a way or integrated into the kits. There is solutions for all of those challenges that we usually get with environmental samples. Now, if you're looking to extract a lot of samples, or you want to automate the extraction of stool samples, there is the possibility of using the magnetic bead kit, magnetic bead-based kit for food. You can use that for stool samples. This can be used by automated extraction using the Kingfisher or similar platforms, or you can also use magnetic separators that are available from McCurry-Nagel for manual separations. If you are more interested in the RNA from stool, there is also a similar kit, NucleoSpin RNA Stool kit, that will then allow you to isolate total RNA. Now, I just want to highlight that in isolating total RNA, contrary to competitor kits, you are also able to isolate the small fractions of RNA within that composite sample. So it's not just your large RNA, but also the small RNA, if that is what is of interest to you. The kits come with a DNAs included, so you don't have to acquire that separately, and they are suitable, as I said, for several sample types. And you get, again, high quality, purity RNA for downstream processes. This is just also, again, looking at comparing the MN kit with other kits out there. I won't tell you what kit Q or kit Z are. I'll leave that for you to guess, but you can see that consistently MN kits achieve high RNA yield across board, even when you look at the CP values following a QRT PCR. Okay, so we're moving on now to the next category of samples, just to look at what are the solutions that are available from our portfolio for you to address your challenges and also to be able to get NGS ready nucleic acids. And when we're talking about nucleic acids in water, generally they come from, it's not just one category, there's two categories of organisms. You have your macroscopic organisms, and you then have your microorganisms. And this will then consist of two different groups of nucleic acids that are usually of interest. So in the later part of this section, I'll talk about the environmental DNA, but initially we'll just talk about how to isolate and what are the interests in isolating the total microbial nucleic acids from water. So when you're isolating total microbial DNA or RNA, usually you are in the space that is concerned with water safety or air conditioner safety, as well as looking at the environmental impact of things like farm effluents and wastewater, or you just want to study the microbiome. So those are usually the areas of focus for people who are looking to isolate total microbial nucleic acids. For this, McCurry-Nagel offers the NucleoMag DNA RNA water kit. So this is a recent product that came out last year, and it's magnetic bead based. Again, if you see mag, it's magnetic bead based, and it is suitable for isolating samples of up to a liter. So we know that if your sample is really diluted, then you want to have high volumes of samples in order to be able to maximize yield to fulfill the requirements for library preparation in NGS experiments. So we are able to do that, and you can also have the flexibility if you're really working with very concentrated samples to use smaller volumes. Again, it's automation friendly. And how it works is that your sample gets filtered, and then your filter is chopped or cut into pieces. You put it into the tubes, the bead tube. This kit, like I said in the beginning or somewhere along the presentation, does not come with the bead tubes, and so you have to get your own. You have to get a separate bead tube, which is available, by the way. So you can buy bead tubes separately. And once you put in your pieces or fragments of the filter and you homogenize with your lysis buffer, then you can carry on with binding of the nucleic acids onto the magnetic beads and just washing away contaminants and eventually eluting your pure nucleic acids. So if you're interested in both DNA and RNA, there's a protocol for that. If you're just interested in either, there's also a protocol for that. So at some stage, you may need to include a DNAs or RNAs in order to specifically isolate one or the other. So it's a very simple workflow, and you can do this on an automated platform, or you can use the manual magnetic separators. So this kit offers you high flexibility in terms of the different types of filters that you can use, the sample volume that you can use, as well as the output of nucleic acid. So you either get DNA, RNA, or both. And it can be automation friendly, high throughput, and you can also work at room temperature. Environmental DNA is usually of interest when you want to track animals like rare species, invasive species, or looking at range expansion or loss, or if you're just interested in studying species diversity, ecosystem health, or discovering new species in the environment. When compared to traditional methods for carrying out such studies, eDNA studies are easier, less invasive, more precise, and offer higher sensitivity. However, the challenges that come when you're trying to study eDNA from water is that most of the time people face DNA contamination, so contamination of your sample with DNA that was not part of the sample. And then also there's a need for solutions for diverse types of water samples, some of which would include inhibitors. And in the past, people have been using also the total microbial DNA RNA kits to do isolation of environmental DNA, which usually is not very suitable. Other people repurpose your tissue kits for this as well. And there's such variety of home-cooked workflows. So for this reason, quite recently, a few weeks ago actually, McCarry-Nagel released the NucleoSpin Environmental DNA Water Kit that allows fast purification of environmental DNA. It minimizes DNA contamination by employing ethylene oxide treated columns, and it's compatible with diverse types of water and filtration systems as well as laboratory setups. Again, there's a range of volume, different types of samples. You can also have the choice between working from filtration as well as working by precipitation of your nucleic acids. More on inhibitor removal as well in this kit. So this is just a brief show of how the filtration workflow works, similar to what we saw for the DNA water kit. And you get your samples, your nucleic acid in under 70 minutes, so it's quite fast. So this kit has been used in tracing toads in Germany. As you can see here, using toad-specific primers, they were able to show that the toads become available or active around about April, May, which was quite interesting for the study. Okay, so now we have our RNA and our DNA, and we want to proceed with storing away for future or downstream processes. And just generally people freeze away the samples, but also just take note that if you're working with RNA, it might be a good idea to convert into double-stranded cDNA, which is more stable, or also to use rotational vacuum concentration that will make your samples more stable over time. And for this, there's a compact benchtop mini concentrator from Madsen Christ that allows quick sample preparation without sacrificing the gentle handling of your samples. Great, so we've come to the end of this presentation. I hope that I've highlighted to you how NGS can be useful for environmental studies. Also how McCurry-Nagel addresses precisely the challenges that are usually faced when working with environmental samples, especially in NGS workflows. So we achieve high purity, high yield DNA or RNA from across a range of environmental sample types. McCurry-Nagel does this best because they've been around for a long time. They have the know-how, they have a R&D section or arm that is dedicated to keep pushing the boundaries, maintaining the standards of quality to provide you with high value end products. So if you don't remember anything I said today, just keep in mind that in preparing your samples for NGS, you must pay attention to the pre-treatment, because that is really where you either make or break things. You either gonna get your samples fulfilling the requirements for NGS library inputs, or you have to start again. So please pay attention to that when you're choosing your extraction methods. Pay attention to the sample disruption, the optimized buffer systems, as well as the strategies for inhibitor removal. And with that, I'm gonna ask you again to please go into the chat box for the fourth poll question and just tell us if you are interested in environmental DNA. So if you're interested in environmental DNA and you want to know more about the new kit that was released a couple of weeks ago by McCurry-Nagel, just tick there and we'll be in touch. And now I'm going to ask Dr. Ogola to assist me in taking your questions. I don't know, I can't yet see the chat, so I'm going to end the slideshow and then stop sharing for a bit. And then I'll join you in the chat box just to see what are the questions